Our genetic disorder project is Kat6a syndrome. We're understanding the genetic and clinical aspects. Okay, overview of the Kat6a syndrome. Kat6a syndrome is a genetic disorder characterized by various developmental and neurocognitive challenges due to mutations in the Kat6a gene. What is Kat6a syndrome? Kat6a syndrome is a rare genetic condition resulting from mutations in the Kat6a gene, impacting brain development and function. This syndrome is associated with distinct physical, developmental, and cognitive impairments that can vary widely among affected individuals.
Also known as the online Mendelian inheritance in man number for Kat6a syndrome is 601408. Other names or abbreviations. Kat6a syndrome is also known as Kat6a related disorder and also known as autosomal dominant intellectual disability, craniofacial anomalies. Golly, all these freaking, all these dashes, bruh. Other names or abbreviations. Kat6a syndrome is also known as Kat6a related disorder and also known as autosomal dominant intellectual disability, craniofacial anomalies, cardiac defect syndrome. Its abbreviation, Kat6a, refers directly to the gene responsible for this syndrome, highlighting its genetic basis and etiology.
Mode of inheritance. Kat6a syndrome follows an autosomal dominant inheritance pattern, meaning one copy of the mutated gene from an affected parent could cause the disorder in offspring. This mode implies variable expressivity, leading to a range of symptoms even among individuals with the same genetic mutation. Chromosome location. The Kat6a gene is located on chromosome 8 at the AP12 region. Variations in its location can influence gene expression and the severity of the syndrome, underscoring the importance of precise chromosomal mapping in genetic research and diagnostics. The Kat6a gene spans approximately 122,000 base pairs in length. This extensive size contributes to the complexity of gene functionality and potential mutation effects, offering challenges in understanding its full range of biochemical roles in human development.
Is that hyperpigmentation?
Protein name and function. The protein associated with Kat6a gene is lysine. It plays a critical role in modifying histones, impacting gene expressions and chromatic dynamics, thereby crucial for development in cellular processes. Effects on cells and structures. Mutations in the Kat6a gene leads to structural changes in cellular components. This impacts neuron development, potentially explaining various neurological symptoms associated with the syndrome. Nature of the defect. The Kat6a gene defects are primarily mutations, including deletions and substitutions. Such mutations disrupt normal gene functions, leading to a spectrum of phenotypic outcomes observed in patients. Go.
Clinical manifestations of Kat6a syndrome. Kat6a syndrome presents a unique set of clinical features with varying prevalence and symptoms among affected individuals. Symptoms. Common symptoms include developmental delays, intellectual disability, dysmorphic features, and various neurological issues. Additionally, some affected individuals may present with heart defects and skeletal abnormalities. Treatment options. Treatment for Kat6a syndrome is symptomatic and supportive, focusing on therapy for developmental delays and speech problems. Early intervention and personalized care plans significantly improve the quality of life for affected individuals. Prenatal and carrier detection. Current prenatal diagnostics for Kat6a syndrome include immunosynthesis and chronic villus sampling. These tests can identify genetic anomalies early in pregnancy, providing crucial information for expecting parents regarding potential outcomes and reproductive decisions. Genetic versus environmental factors. Kat6a syndrome is primarily a genetic disorder caused by mutations in the Kat6a gene. However, Ongoing research investigates whether environmental factors, particularly nutrition, can influence symptom severity or presentation in affected individuals. Potential for gene therapy. Gene therapy holds promise for Kat6a syndrome by potentially correcting mutations or compensating for defective gene function. Ongoing clinical trials are exploring innovative approaches, including CRISPR technology to restore normal gene activity and improve health outcomes. Future research directions. Future research on Kat6a syndrome aims to better understand the molecular pathways involved and to identify additional mutations that may contribute to the disorder. Collaborative efforts in genomics and proteomics are essential for developing effective treatments and management strategies. And that concludes our presentation on Kat6a syndrome.
I might swerve in that corner, whoa, whoa. Hold on tight, cause I tweaking this, I let go.